Hey guys, my name is Ishaan and today's video is going to be something quite different. Never done it before, so I'm going to be trying to do an A-level biology paper. Even though I'm a GCSE student who's just going on to do A-levels, I've got no knowledge of the biology A-level course, and this video is basically going to be me doing revision for the first 30 minutes. Again, it's going to be sped up, so you're just going to be seeing me revising like the first topic of A-level biology, which I think is cell structure or something like that. I'm only going to have 30 minutes to revise, and then I'm going to have to do a physics and maths tutor paper. Again, this is not intended for people who are looking to revise A-level biology. This is just a bit of entertainment. See how much I remember, see how much I can actually do, and what I actually get at the end. So let's get into it. Let's start revising for the first 30 minutes, and then we'll go on to do the paper. Let's have a go at this paper. Yeah, biology students don't come at me because I don't know what I'm really doing. Smooth ER, smooth endoplasmic. Smooth, I remember, okay, so I definitely know this because smooth, oil is smooth, therefore oil is lipid, so lipid synthesis. I'm not going to write it in full just to save time. Rhizome, that, digestive enzymes. It contains digestive enzymes. Contains digestive enzymes and breaks down cell. Ribosome, that's easy. Protein synthesis. Okay, one theory that about organelles here. This theory suggests chloroplast cells need every bacteria in larger cell. The following describe chloroplasts. 3 statements used evidence for them. When the hell have I learnt this? Chloroplasts contain ribosomes that are smaller than those. Yes. Chloroplasts contain, but why would that? No. Chloroplasts are similar size to bacteria. Similar size to bacteria. Yes. The inner membrane of mitochondria is folded to form a maybe chloroplast. Maybe chloroplasts have their own. They do, so yes, that. Name 2 features of a plant cell that are not features of an animal cell. They contain chloroplasts, and they also have cell wall. Name 1 structure present in animal cells, not present in. Oh no, I don't know this. Is it lysosome? probably wrong, but I don't know. Describe the roles of the cytoskeleton. I know this, I think. So, what does it do? Helps support cell. Responsible for movements of substances in/out cell, movement of flagellum, I think, or cilia. Flagellum is the tail, and the cilia are the like sweepy cells. What else does it do? Cytoskeleton. Oh, movement of chromosomes. And that's all I remember. Hopefully that's right. 3 marks. Okay, pancreas is an organ that secretes protease enzyme. Outline organelle. So basically, this is asking us to the protein thing. So, oh no, I've forgotten it. Okay, let's just do easy mark. Ribosome, where protein made. So where protein synth occur. And let's say is that a transcription? Transcription occurs, and at the nucleus, translation. No, I think this is the other way around. I don't know. Ribosomes are okay. Then on this rough endoplasmic ER, it folds and processes proteins. I don't know. Then vesicles to the smooth ER, where the proteins are processed and packaged. Proteins are processed and packaged. Then they move into vesicles. Then the vesicles, I think they fuse to the what's it called, cell membrane. No, not cell membrane. What's the word that is it plasma membrane? Hopefully it is. Hopefully I got 3 marks there. Name components A and B. A is the nucleus. Was that the nuclear envelope? That's the nuclear envelope. E, and that's the nucleus. And those are the pores. I don't know. B is chloroplasts. State function C has to be mitochondria produce ATP. I'm not going to write in full sentences. D is smooth ER. So smooth ER, oil is smooth, lipid is oil, so lipid synthesis. Please tell me that's right. Student detail component C not clearly I don't know about this. I forgot about light microscope because of the light. I don't know. Staining is a process often used in microscopy. Describe advantages of staining. Okay, well, staining, I think this is common sense. Becomes more visible. So you're able to distinguish different organelles. Some organelles are transparent, and then becomes more contrast is seen. Hopefully that got 1 mark. Okay, place a tick or cross in the box to indicate whether this feature is present or absent. Okay, this looks easy. Chloroplasts, yes, they are in plant cell, not in animal cell, not in bacterial cell. Cellular cell wall, that's in plant cell. Animal cell doesn't have a cell wall, bacterial cell doesn't. Centrioles, plant cell, I don't think it has. Animal cell, I think it does. Bacteria, does it? No, hopefully not. Ribosomes, I think everything has it. Okay. Investigator cells were broken up and then components separated. Tubes were tested, determined identity components, ability. Okay, so identify the tube that contains the following organelles. Nuclei. Nuclear presence of DNA, trace amount. It has to be 1. Ribosomes. Ribosomes, is that proteins? Yes. No proteins, no proteins, no proteins. It has, oh, so 3, 4. Has to be 4. Mitochondria, ATP, no ATP. Okay, it has to be 2. And lysosomes, oh, back to digest, so it has to be some of it, so it has to be 2. 4, 2, 1, 4, 2, 2. Okay, which organelle is listed smallest size? It has, so ability to make ATP, that's mitochondria, that's like that. Presence of DNA, that's the nucleus. Proteins proteins are tiny, so no, ribosomes are tiny. Bacteria has to be lysosomes, so I think it's ribosome. Okay, hopefully that's right. C, D, and E. Name C. C has to, so that's a Golgi. Oh no, I forgot to talk about the Golgi. What does the Golgi do? Oh no. C is the vesicle. Oh, I got the previous one wrong then. D is, what's D? Where's D? D is the plasma membrane. And E is, what is that? Is that not the rough ER? It's pointing to the ribosome. So just type one type of extracellular protein secreted at B. What's B? So that's enzyme. Enzymes. But what enzyme? Protease? I don't know. Hopefully. So there's one stage secretion of protein. Okay, so probably fusing with plasma membrane. What fuses the vesicle? Outline the role of the Golgi apparatus. Oh no, I don't know this. Is it just to process and repackage proteins into vesicles and then fuse with membrane, plasma membrane? So that means I got the one up there wrong because I didn't talk about the Golgi. So maybe maybe vesicles, no, maybe I got this wrong, that's wrong. So, then that's the Golgi, is where proteins processed and packaged into vesicles. Maybe that's right, I don't know. Then where am I? Identify 2 features visible which would not be present in prokaryotic cell. Oh, 2 features visible in 2.1. The Golgi, and this is a complete guess. Golgi, ribosome has to feature, so that's out of question. And the rough ER? I don't know, please. Name one not in the cytoplasm that would be that is not found. Is it free DNA or free genetic material? I don't know. Okay, student wants to observe some red blood cells under a microscope. The student placed a small sample of blood onto a microscope stand and added a drop of distilled water. When viewed at high power, the student observed the red blood cell had burst. Yeah, because of osmosis. In a similar epidermis, the student did not burst. Okay, I remember doing this in GCSE, but it's a 5 marker. That's not what we have to do. So let's just try doing this. So a red blood cell has no cell wall. No cell wall. And it has a low water potential inside and high water potential outside. Now water is drawn in via osmosis. So it basically swells, and then what happens is it bursts because of due to high pressure. Now the plant cell has a cell wall, hopefully. Has cell wall. So it becomes turgid, or is it flaccid? Becomes turgid. What's flaccid then? I don't know. So it becomes turgid, and so cell wall is strong. Cell wall is strong, and that means not burst. Okay. So just how the student could modify the procedure to observe red blood cells without them bursting. How would you modify it? So it has to be something to do with water. So not use water or salt water, or what's it called? What's the what's the solution? The like eye solution. Say saline. Saline. Okay, name the mechanism by oxygen enters red blood cells. Diffusion, easy. State the process by root epidermis cell absorbs minerals from the soil and describe how these cells are specialized. Okay, well that's active transport. So they are thin cell wall, so short diffusion distance. Large surface area because of the extensions. Describe how these cells, what else is there? I'm so silly. Oh, mitochondrion, lots of mitochondria. Yeah, lots. Energy, ATP. What's the other? Carrier proteins. Lots. I don't know. Hopefully that gets me 2 marks. Please tell me that's the end finally. Okay, well, let's mark that and see how we did. I don't know what it's out of, but we can calculate that. Let's go to the website. I've just opened my Mac. Google Physics and Maths Tutor mark scheme. Okay, let's have a look at the first one. Okay, so the first one should be transport of lipids, accept processes toxins. So makes lipids. Oh, please tell me that's right. Let's just mark it as right. Lysosome, it contains, yeah, I got that right. And ribosome protein synthesis. I got 3 out of 3. Good. The next one should be taking the first, taking the second, taking the last. 3 out of 3. I'm on a roll already, 100%. 2A, this was easy. Cell wall and chloroplasts, that's 2 marks. The next one is centriole. Don't know. Oh no, except lysosomes. Never mind. That's right. 1 mark. Support role of the cytoskeleton. So whole cell support. So cell support. So that's 1. Oh, that's 1 mark. Movement of phlogia. Yes, movement of phlogia. Another mark. Movement of chromosomes. Another mark. So that's 3 already. I'm actually proud of myself. This is where I think I messed up. Okay, so nucleus contains gene transcription. Nucleus. Oh no, I got that messed up. It's transcription. So C for, okay, and that's translation. So nuc for transcript. So ribosome is where protein synthesis. I got 1 mark at least, 1 there. Vesicles for transport of protein. Vesicles. Golgi protein. Okay, that's another mark. Vesicles fused to plasma membrane. That's another mark. Vesicles for transport. Oh, I didn't say transport, but I did say vesicle. Let's just give myself a mark. I probably didn't get it, but let's just say I got 4 and probably 3. Yeah. Question 3. Nucleus and chloroplast. Good. 2 out of 2. Mitochondria produce ATP. Yes. And lipid synthesis. Transport processes. Yes. Lipid synthesis. So I got 2 there. So there. B, this is what I did not get. Let's just, so resolution not high. So I got 0 there, that's sad. C, makes it visible, that's 1 mark. Increases contrast, more contrast? Let's just, yeah, so 2 marks there. Question 4. Okay, so tick, not, correct. Tick, not, tick, not, and tick, tick, tick. Well, I'm actually doing so good, 4 marks. Okay, next one. I think I got 1, 4, 2, 2. 4 marks there. Ribosome. I'm actually on a roll. I'm so happy. Okay, C is the vesicle, correct. Plasma membrane and ribosome, 3. Yay. Enzymes. Enzymes. Except enzymes, but it doesn't come up with protease. So let's just give myself a mark because I said enzymes. So that's 1 stage during transport vesicles to plasma or fusing vesicle to membrane, fusing with plasma membrane. I think that's a mark there. Outline the role of Golgi replicator. Receives proteins from ER, so I didn't say that. Modifies protein packages. So processes and repackages proteins into into vesicles. I think that's 2 marks because process. Yeah, so let's just, and then it's not fused with it. Okay, I need to remember it makes lysosomes. BI, identify 2. Nucleus and nuclear, ribosome, okay, Golgi, that's 1. And rough ER, that's another. Yay, 2. Free circular DNA, that's correct. Okay, I don't know about this. You sort of use my... So cell has a slower water potential than distilled cell. The cell has a slower water potential. The low water, I don't know. Let's just say that's 1 mark there. Water is drawn by osmosis, that's 1 mark there. Ideal cell surface, weak, cannot withstand. Swells, bursts. Okay, I think that's another mark due to high pressure, 1 mark there. Plant cells strong, that's 1 mark there. Becomes turgid, 1 mark there. So there's 4 maximum marks I can get. No, 6 marks that are available, 4 marks I can get. 1, 2, 3, 4, 5. Did I get 4? 5? Okay, no. QWC, 2 technical terms used in context spelled correctly. Well, I have to spell them. Okay, so yeah, I think 5 then. Because I did use osmosis, which was 1, and turgid, which was 1. Okay, next one. Please tell me this is right. Salt. Except saline. Good, yay. 1 mark there. Diffusion, 1 mark. Okay, active transport, that's 1 mark. Thin cell wall, that's 1 mark. Large surface area, that's 1 mark. More mitochondrion, lots, hopefully that's a mark. And carry proteins, that's another mark. Okay, so that's, let's say I got 3 there. And that, I don't know why, that felt easy. I don't know, let's just count it up. I don't have a calculator, which is annoying, but let's just use the one on my laptop. Calculator. So it's out of, so let's just do 10. So what's it out of? 10 plus 10 plus 10 plus 9 plus 8 plus, no, I've been doing something wrong on the calculator. No, let's go from the beginning. 10 plus 10 plus 9 plus 8 plus, no, I'm doing, oh no, what am I doing? No, I'm so silly. 10 plus, no, I can't use a calculator. I just can't do it. This is so annoying. 10, 20, 29, 29 plus 8. 29 plus 8 equals 37. 37, 48, 49, 50, 51, 52, 53, 54. 54, is it out of 54? Let's just do it again. 6 plus 11 plus 8 plus 9 plus 9 plus 10 plus 10, 54, yeah. And what did I get out of 54? So 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 48, 49, 50, 51. 51, what the hell? I'm gonna assume that's good. 51 out of 54. Is that good? Anyone? Did I actually get it right, or was I like ticking it without knowing I got it wrong? If anyone has any advice, please let me know, because I don't know if I got it all right or wrong. Like these, I don't know, I might have been a bit generous. Please let me know if you guys do A-level biology. This was sort of just like a random upload, seeing if I could do this A-level paper with only 30 minutes of revision. I guess I could, but it wasn't that. It was the first topic, which was actually not that bad. Let me know how I did compared to how you guys are finding it. But thank you so much for staying till the end, finding out what I got. I'm pretty proud of myself. If there are any A-level biologists here, please let me know if I did it right or did it wrong. But promise, that was the first time I've ever done it. Yeah, thank you so much for watching. Please subscribe if you haven't. Have a great day. See you in my next upload.